Hello students, now we are going to carry forward the work of the practical of isolation and identification of lactic acid bacteria. Here we are getting the result in our plates. I will show you one by one the results. In case of MRS plate having curd, we have two separate colony of lactic acid bacteria. In case of milk, we are getting nice result, and in the sector 1, 2 and 3 also we get lactobacillus. In case of cheese, we are getting some little contamination and also the growth of lactic acid bacteria. Okay, and in case of glucose yeast acetic acid agar, the growth is very scanty in all the three different natural samples. So I think that it should be incubated for longer time period, and maybe because of the fluctuation of the temperature the colonies could not grow properly. Okay, now the very first thing we are going to do will be, we will take the best plate from all of these three plate of MRS media. The best result is seen in case of the milk sample. So we will select one particular plate of milk as a natural sample here, and now we will proceed further in the detailed manner. Now, how to identify the colony of lactobacillus? Lactic acid bacteria are many. There are so many different genera, but here we are having interest only in lactobacillus. So lactobacillus will give us very smooth and white colony. It is usually white, that is creamy white or yellowish white, and it will give you smooth appearance, and it may be little bigger in the size, not so small, because 48 hours are already completed. So I can see several colonies here in the second sector, 1, 2, 3 and 4, and out of these 4, this is well isolated and very nice to work with because it is separated and well isolated. So I am going to use this particular colony. Okay, now before I go for anything, I will just remove the vapor. So for removing vapor, I need the cotton. So now the colony will be more clearly visible. I will keep it in the sunlight and try to see the colony. Now I will write down the colony characteristics in my record. Okay, so I will start with the size of the colony, shape of the colony, margin, elevation, texture, opacity and color. Now let us discuss about all these criteria for lactobacillus group of organisms. Okay, so here the colony is big in size, the shape is round, opacity is, it is opaque, non-transparent, margin is entire, it is very smooth in appearance, the texture is very smooth, elevation is slightly raised, and color is dirty white. So this is about colony characteristics, and it has to be written before we use colony for some other purpose. Now we will have three different things which we are going to perform now from this colony. First of all, we will try to check whether it is catalase positive or catalase negative. Then after we will make the suspension, and then after we will go for the gram staining, and also for some special stainings like capsule staining, spore staining, and metachromatic granule staining. Now here, very first thing we will do will be, we will sterilize our nichrome wire properly, and then after we will take out little material from the colony and keep it on the center of the slide, and then after we will add one drop of H2O2, and we will try to see whether catalase enzyme is positive or not, whether we are getting the bubbles or not. So that is our now lookout, and that we are going to do now. And by the time we will also make the suspension of lactobacillus. So now we will allow it to cool down. It is now already cool. We will use it for taking out some material.